Welcome to this lecture. Today we would be discussing about DNA fingerprinting. The main learning objective of this lecture is you will be getting to understand what is DNA fingerprinting, what is the technique of it, what kind of sophisticated techniques we use in this technique, and what are the case studies pertaining to this lectures, and what are the applications of DNA fingerprinting technique. First of all, before starting on to this lecture, we should first of all must discuss what is DNA fingerprinting actually. When this technique was not there at that time, what the scientists used to do, they usually, they go on for checking skin fingerprints. That means 100 years ago, scientists discovers that patterns that are present on the onto the fingertips of the skin that can be used to establish a person's identity. At that time, this technique was basically used. This was the conventional method, as you can see, this fingerprint. If some murder is happening, or so, if some sort of dispute is there, in that case, this was the main thing. This fingerprinting was the main thing that were, mostly you can say mostly the scientists used to do, or mostly the police used to do. So in the year of 1895, there was a English genetist, Alec Jeffreys. He was studying at the University of Leicester, and he basically discovered that there is some inherited variation within the genes, and because of that, each individual have a different sort, or you could say a different kind of gene clusters or gene groups. So we discovered a new kind of fingerprint out of these variations in the gene. And this unique signature that is found in each individual and that varies as per the genetic makeup. He reported this thing as DNA fingerprint because our main thing is it is basically dependent upon the variation of the genes. So in short, what you can say, what is DNA printing, DNA fingerprinting? Every individual they carries a unique set of genes. That means chemical structure of the DNA is same, that it has four nitrogen bases, it have sugar, it have phosphate group, but the order of base pair differs between each individual, and each of the cell it contains a complete set of DNA that identifies the organism as a whole. And however, out of the total amount of the DNA, only one tenth of one, or you can say 1/10 of 1% of the DNA differs from person to person. So, depending upon that variation, they discovered this technique known as DNA fingerprinting. Now, this DNA profiling, it depends upon a small portion of the genome. That means exon code to exons that code proteins. That means, as we know that eukaryotes are the genome of eukaryotes. It contains exon and introns. Exon will code for proteins, and introns do not code for any kind of proteins. However, in this introns, they contain certain repeated sequences which we call it as VNTR. That is variable number of tandem repeats. Basically, it is a repeated sequence of length around 1 to 100 base pair. And there are many kind of VNTRs that are present in a particular individual. Some of the VNTR we inherit from our mother, and some we inherit from our father. So, this is the actual concept actually. So this is the homologous chromosomes, repeated sequences of VNTR sequences shown in the red color. And this is maternal, this is paternal. And when you will, analyze these sequences, analyze these VNTRs by aggressive electrophoresis, you will be observing the bands at different locations. So suppose there is some you can say there is some forensic work happening. In that case also, the forensic sample F, A, B, C, that means you have to compare the homologous sequences, and you have to compare for the VNTRs that are present, and depending upon that, when you run it by the same number of VNTR, same size of VNTR, presence of these VNTRs in different individuals, we can ascertain the susceptible person, or we can uh, go for the detection of uh, main culprit. So, what is DNA fingerprinting actually? That means DNA fingerprinting, it is not only restricted to the detection of microsatellites. That means 1 to 6 nucleotide repeats that we are talking about VNTR that are completely dispersed throughout the chromosomes. If we want to go for the detection of these microsatellites, we have to design certain probes. What are probes? Probes are short stretches of nucleic acids. These probes are basically used to identify the microsatellites surrounding the specific microsatellite being analyzed. And these kind of microsatellites, that means 1 to 6 nucleotide repeat sequences, we call it as short tandem repeats or STR. And these STR are basic, or you can say the foundation of this DNA fingerprinting technique, because the FBI has chosen 13 unique STR that they use for fingerprinting for testing, and that complete system, that complete 13 STRs are coded, or you can say they are mentioned in CODIS system, that is combined DNA index system. Whenever some crime is happening at that time, that CODIS system is basically used. Now, what is the technique actually? DNA fingerprinting technique. There are two main type of DNA testing. One is RFLP, that is restriction fragment length polymorphism, and another is polymerase chain reaction. If you talk about restriction fragment length polymorphism, in that case, as the name suggests, restriction fragment, that means we have to use restriction enzyme so that different length fragments can be generated, and depending upon those fragments we can ascertain the polymorphism. So for this very technique we require very large amount of DNA, and DNA shall not be degraded. However, another technique is PCR reaction, that is polymerase chain reaction, in which a very small amount of DNA is required. Even if it is partially degraded, even then our work will be on. However, this technique, it suffers from a major disadvantage, that contaminating DNA. It is, if some contamination is there, in that case our results may be wrong. Next is, suppose you want to go for this DNA fingerprinting technique, then what are the steps? Then the first step is specimen collection. That means, first of all, we need to understand what are the sources of DNA. In that case, for this very collection, we require certain precautions that we have to take. That is, we have to wear disposable gloves, and you shall not repeat it. That should be one-time use. Then you have to use disposable instruments. Don't talk, sneeze, or cough around the sample site. Avoid touching any item that might contain DNA. So, air dry the evidence before packing. Otherwise, if the some moisture is there, in that case, mold may develop and it may contaminate the sample. So there are certain enemies of evidence, that means that can destroy the evidence before the testing is done. So that is sunlight and high temperature, bacteria and moisture. Because if some bacterial contamination is there, even then your uh, uh, result would be right. If some moisture is there, in that case mold may develop. If the sunlight and high temperature is there, and if something is sensitive to heat, in that case that evidence may be destroyed. So DNA fingerprinting, it is a comparative process. That means you have to take the samples from the crime scene and you have to compare it with the suspect DNA. And the best sample that you can take if you want to go for DNA fingerprinting, that is fresh blood, whole blood. So sources of sample: blood that is maybe extracted dry or wet, that means WBCs. Buccal or cheek cells, it is very easy to obtain. Semen, dried, can be analyzed years later. Hair roots, saliva, skin cells, these all can be the sources of sample. Next is step 2, DNA extraction. Now, after collection of the sample, next step is the extraction of DNA. That can be, you can do the isolation by using many methods, either chemically or mechanically. Mechanically means you have to use glass beads so that you will be using the force. Third step is after doing the collection and DNA extraction, next step is RFLP. In that case, we will be using restriction enzymes. So the nucleotide sequence variation in the region of DNA, because of these variations, different sized fragments would be obtained whenever you will digest it with different kind of restriction enzymes. As you can see, these are the sites. When you will cut, this is the cutting site actually, this position, and these sticky ends are generated. In a similar fashion, if this length, if in a kind of individual, if this is not the site or this site is not properly there or there is some sort of mutation, in that case, different fragments would be generated. Next is after getting the RFLP done, next step is treat for this very complete step. First step was restriction enzyme digestion. That means restriction enzyme will cut the DNA at restriction sites. You can use range of restriction enzyme, whether they are sticky end cutter or they are blunt end cutter. Whether you can use one sequence or you can use more than two sequences. That is, you have to design your experiment accordingly. After that, once the restriction digestion is complete, the next step is agarose gel electrophoresis to separate all these fragments depending upon the variation in the size. Once the complete thing is separated, the fragments are there, then you need to treat your gel with some chemical or you can heat it so that the DNA that is present in there, that is in double stranded form, it shall get denatured, because once it is denatured, it will be single stranded. When you will be adding the specific probe, that single stranded probe will attach to the single stranded DNA. So this is the step actually. This is the DNA. We have separated the DNA onto the agarose gel, transferred or blot the DNA fragments from gel to membrane. Then membrane with DNA transferred over it. Then you need to incubate this with the probe. That means from here we would be proceeding on to the southern hybridization that we discussed yesterday. Transfer the DNA fragments from gel to nitrocellulose membrane. Incubate the membrane with the probe, that is a short stretch of complementary DNA which is tagged with a radioactive tag. Then once this complete transfer and probe hybridization is complete, you need to expose this thing to the X-ray film so that you can get the results. So such kind of bands would be observed. So this is the complete process of RFIP. This is the blood sample. Then you have to take the DNA sample. You have isolated the DNA, subject it to restriction digestion, separate the fragments. Then the southern blotting would be performed now. Transfer these fragments onto the membrane. Then hybridize it with the radioactive probe. Excess of the radioactive probe would be washed once the incubation is over. Then develop the X-ray film to detect the radioactive probe, and you would be getting such kind of bands. Once this is done, we can also use polymerase chain reaction technique, which is used to amplify DNA that is found at the crime scene. So if the amount is very less, in that case, we can go for the polymerase chain reaction. In that case, we go for STR analysis, that is short time repeats. As I told you that there are 13 STRs that have been reported by FBI. So you can basically go on for amplification of those STRs. So, as I told you, there is very small amount of DNA available for the DNA fingerprinting. In that case, we will be going for the PCR technique. This is the target sequence. You have to design the primer complementary to that very STR, and new DNA would be synthesized. And this complete thing is exponential, so you would be having million copies of that very particular STR. So once we have isolated the DNA and amplified it with PCR, next you can proceed on to digestion with restriction enzyme. Then different size fragments could be performed, and you can go for the RFLP technique so as to detect the bands. So these are some case studies pertaining to this complete concept, that how the even after the discovery of this very technique, this sophisticated technique, people are using some other means so as to protect themselves. So this is the first case study, fake DNA evidence. There was a Canadian doctor who was basically charged for sexual assault in the year 1994. And what he does, he gave the blood samples. And at that time, what happened when they took that blood samples, they found that he is not the culprit. But after that, after the thorough investigation, they came to know what he did. He surgically inserted a pen was drain filled with the foreign blood and anticoagulants in his arm. So when the when the forensic team was taking the sample, the foreign blood was taken out, not his blood. So he was basically faking with the DNA evidence. And this thing came to the light when police obtained a sample of his hair. At that time, the complete bands that were obtained after DNA fingerprinting, they were different in case of blood and different and in case of hair. From there, they ascertained that he forged the thing there so as to protect himself. So he was convicted in the year 1980. Then you can use this DNA into use, because when there was in the December 9, 2004, there was South Asian tsunami, and at that time around many peoples and that have claimed many lives of at that time. And for the identification of those people, the main scientists were into that they use mitochondrial DNA and YSTR analysis, and within 3 months, 800 victims were identified and they were handed over to their families. So the main thing is the this technique is very helpful in ascertaining or in helping many things. For example, in terrorism and natural disaster flows, new technologies can be developed. When the World Trade Center was hit in the September in the year 2001, and that time also DNA technique was used to identify the remains of the victim. Because after the blast, you can't ascertain that which of the persons were there. So depending upon their remains, the DNA was isolated, and this DNA fingerprinting technique was used to ascertain the identity of victims. That times there was large amount of debris, heat, microbial decomposition of remains, because hundred and thousand of tissue samples were obtained, and those samples were basically identified by this DNA fingerprinting technique. So the power of DNA fingerprinting is immense. You can observe same thing in case of some another case that was Nicole Brown Simpson and Ronald Goldman crime scene, in which both RFLP and PCR technique was used, and in which when they performed PCR, when the 3 drops of blood were tested, then it was found that this blood is basically of Simpson. But when 2 drops were tested, at that time, drop 1 and drop 2, they were completely showing the different result to someone other than the OJ Simpson sir. So application is immense. That means you can go for the detection of diagnosis of disease. Suppose if there is a normal individual, this is a diseased individual, this is a career. So depending upon the DNA profiling, you can ascertain that which one of them is career. Because this complete, that means inheritance of a particular disease can also be ascertained by using this DNA fingerprint. Paternity testing, if there is some dispute regarding the male identity of biological father or mother, in that case also we can go for the DNA fingerprinting technique. We can go for checking by using samples from father 1, father 2, mother and child, and by comparing the bands we can ascertain that yes, this is the biological father or mother of a particular child. Paternity testing, that means we can analyze the sample from the child and adults involved. We can use mitochondrial DNA if the samples cannot be analyzed by PCR or RFLP, because all of this mitochondrial DNA it is inherited from mother. That means the particular kid inherit that thing from the mother. So and due to some random mutation, the sequence may vary, but the rest of the things remain same. So it is very useful in case of forensics. because depending upon many crime scenes and many murders or accidents, you can go for using this very DNA fingerprinting technique to ascertain the identity. You can take the samples from jeans, t-shirts, skin samples, or some debris. These are the 13 core STI locations that I told you that FBI has recognized 13 STRs. So these are the 13 STRs actually that are present at the different locations, and these are named as such, yellow one. So by ascertaining this STR, as you can see in case of victim, these are the 13 STR location, the sample that they have got from crime scene and the sample that they have got from suspect. And what they do, they usually compare the presence of these STR between the crime scene sample, suspect and victim. So as to ascertain the identity. In case of genealogy, also it is of immense importance, as I told you. In case of any natural disaster or any kind of uh, uh, mishappening, in that case also to ascertain the identity of victims or to ascertain the identity of persons, the DNA fingerprinting technique is being used. And you can go for the detection that whether a particular disease is more prevalent in a particular kind of individuals or in a particular kind of or it is specific to a particular geographical location. So we can simply go on detection. This is a bit of exercise pertaining to this. Interpret the results of the following DNA fingerprints. Both the daughters named as D1 and D2. Here in D1 and son S1, they share genes with mom and dad. As you can see, these are mom and dad. And this blue band is similar to that of mom. And this yellow band is from dad. This one is also from father and this one is from mother. And this D1 and S1, they share similarities with the mother and father. However, in case of daughter 2 and S2, they are not showing any kind of similarity. However, the daughter too has some genes of mom, but not the dad. And son too doesn't share genes with the parent. In that case, this D1 and S1, that means daughter 1 and son 1, they are the kids of the mother and mom and dad, the biological father and mother of these two kids. However, in case of daughter 2, the biological mother is this very person, but the father is someone else. And in case of son, this kid is not the biological son of these parents. From here we can basically go for detection of many kind of things. However, this technique is very important and very helpful in solving many crime mysteries. So I hope the concept is clear. In case you have any doubt, you can ask.